Reactions of amino acids. Reaction due to carboxyl group. Decarboxylation. Amino acid loses the carboxyl group and gets converted to amine. Histidine undergoes decarboxylation to form histamine, responsible for allergic reaction. Tyrosine undergoes decarboxylation to form tyramine, which precipitates migraine attack in migraine patients. Tryptophan undergoes decarboxylation to form tryptamine, excreted by the intestine. Lysine undergoes decarboxylation to form cadaverine. It is a polyamine responsible for cell growth and differentiation. Glutamic acid undergoes decarboxylation to form GABA, inhibitor neurotransmitter. Next is amide formation, where amino acid reacts with ammonia to form respective amide. Aspartic acid reacts with ammonia to form aspargine. Glutamic acid reacts with ammonia to form glutamine. Next, reaction due to amino group. First is transamination, next is oxidative deamination, and the third one is formation of carb-amino compound. Now, transamination is a process where amino acid gets converted to keto acid and keto acid gets converted to amino acid. The process is as follows. Alanine, the amino acid, donates the amino group to alpha ketoglutarate. The group transfer is done by pyridoxal phosphate. It accepts the amino group and gets converted to pyridoxamine. Alanine gets converted to pyruvate, and alpha ketoglutarate, after accepting the amino group from pyridoxamine, gets converted to glutamate, the new amino acid. Now, the significance of transamination is that it is involved in interconversion of amino acids, synthesis of non-essential amino acids, and the transaminases, especially alanine and aspartate transaminase, are used for diagnosis and monitoring various liver diseases. The next reaction due to amino group is oxidative deamination. Now, this is coupling of transamination with deamination. So, alanine amino acid donates the amino group to alpha ketoglutarate and gets converted to keto acid, that is pyruvate. Alpha ketoglutarate accepts the amino group and gets converted to new amino acid, that is glutamate. Now, this reaction is taking place in all the peripheral tissues, and this glutamate reaches liver, where it undergoes oxidative deamination and the ammonia is released. Simultaneously, NAD is getting reduced to NADH. Now, this reaction is taking place only in liver. So, this coupling of transamination with deamination is also known as transdeamination. Next one is formation of carb-amino compound, where carboxyl group gets attached to the amino terminal end of hemoglobin to form carb-amino hemoglobin. Reactions due to side chains. Transmethylation reaction. This is because methionine acts as a methyl group donor and it gets converted to homocysteine. So this methyl group acceptors include the following. Guanidinoacetic acid gets converted to creatine. Norepinephrine gets converted to epinephrine. Epinephrine gets converted to metanephrine. These are catecholamines which are responsible for increasing the force of contraction of the heart, increasing the heart rate and increasing the blood pressure. Ethanolamine undergoes methylation to form choline. Phosphatidylcholine is one of the component of lung surfactant. Acetyl serotonin derived from tryptophan accepts the methyl group from methionine and gets converted to melatonin, responsible for maintaining the circadian rhythm or the biological rhythm. Histidine undergoes methylation to form methyl histidine. Next is formation of ester formation by OH group. Serine and threonine have got OH group at its axis center, reacts with the phosphate group to form esters, and these contribute to the formation of phosphoproteins. Reaction due to amide group. Aspargine and glutamine react with carbohydrates to form glycoproteins, and the reactions due to SH group of cysteine. Now, cysteine is the reduced form, and the SH group of cysteine, two different cysteine molecules, they are linked to form disulfide bridges, which are responsible for connecting polypeptide chain in secondary structure of the. The next property of amino acid is the isoelectric point or the isoelectric pH. It is at this pH where the molecule carries no net charge and they occur as zwitterion. So, what are the properties at this isoelectric point? So, there is no charge, no mobility, minimum solubility, minimum buffering capacity, but maximum precipitation at this isoelectric. Now, coming to special functions of amino acid. GABA, derived from glutamate, is an inhibitor neurotransmitter. Gabapentin is derived from GABA. It acts as a pain relief agent. Dopamine acts as a neurotransmitter in basal ganglia, derived from tyrosine. Histidine binds to ion of hemoglobin, has got buffering capacity. This is because the imidazole group of histidine has got pK value of 6.1, which is close to physiological blood pH, hence it acts as a buffering agent. Histamine derived from histidine is responsible for allergic reaction. Thyroxine derived from tyrosine is a thyroid hormone responsible for maintaining the basal metabolic rate. Serine is present at the active center of all the proteolytic enzymes involved in digestion of carbohydrate, proteins and lipids. only digestion of proteins. Proline is responsible for creating bend in the structure. Ornithine and citrulline derived from arginine are intermediates of urea cycle. Homocysteine methionine derivative precipitates thrombus formation or activates platelets and precipitates thrombus formation. So this is the quick summary of the property of the amino acids. Reaction due to carboxyl group or decarboxylation. Examples include histamine, tyramine, tryptamine, cadaverine and GABA. Amide formation, aspargine and glutamine. Reaction due to amino group transamination, where amino acid gets converted to keto acid and keto acid gets converted to new amino acid. Then oxidative deamination, where the transamination is coupled with deamination of glutamate, releasing the ammonia which is detoxified in urea cycle. Formation of carbamino hemoglobin. Reaction due to side chain transmethylation, where methionine acts as a methyl group donor to for formation of creatine from guanidinoacetic acid, epinephrine from norepinephrine, metanephrine from epinephrine, choline from ethanolamine, melatonin from serotonin, and methylhistidine from histidine, ester formation, glycoprotein formation, and sulfide bridge formation from cysteine. The last part is the isoelectric point, where the amino acid have no charge, no mobility, minimum solubility, minimum buffering capacity, but maximum precipitation. So this was all about properties of amino